Okay, thank you for joining in. I think we will start with the session and let's see how many of others join. So, very good evening. I am Guru Priya R, a Prime Minister Research Fellow in the Department of Civil Engineering, IIT Madras. I'll be handling this live practice session for the course Applied Environmental Microbiology. So this is the fourth live practice sessions. So this was supposed to be held on Tuesday, but uh, due to some health issues, I was not able to conduct it on Tuesday, and we are having it today. From the next week class onwards, it will regularly be on Tuesdays only. Only this week's Tuesday class was postponed to Friday session. So in this week's course content, it was completely about various microbial ecosystem. I hope the lectures were clear. Uh, you have any specific doubt? Okay, I guess Nikita, you don't have any specific doubt in the course content of this week. Do you have anything? Okay. So I guess I will post the first question for today. Right. Okay. I guess I see two more participants who have joined. So welcome you to the session. And in today's session, we will discuss about microbial ecosystem. So this was the first question, and you may try answering it. Okay, as Nikita has answered it correctly, ecosystem combines abiotic and biotic factors of the environment. So you all know many abiotic factors that are concerned with the environment. Biotic factors, in the sense, they they possess life, or they are life forms like microbes or other forms of living, which may be prokaryotic or eukaryotic, that influence uh, the presence of other biotic and abiotic factors in the environment. So, ecosystem combines abiotic and biotic factors of the environment. This is the second question. Good evening, all. I'm sorry I got disconnected. Is the screen visible now? Okay. So in today's session, we'll go through microbial ecosystem as it was discussed in the lecture contents. So, first question, this was the first question that I posted today. It was ecosystem combines dash and dash factors of the environment. I guess Nikita had answered it right. It was abiotic and biotic factors of the environment. So, ecosystem combines abiotic and biotic factors of the environment. So, the abiotic components of the environment include um, soil, water, etc. And the biotic components include the microorganisms or the plants and the animals that live in the ecosystem. So this was the second question. I guess I was disconnected while I was posting this question. So you can try answering this question. Okay, I'll wait for two or three more responses. So this is right. An ecosystem is a complex of multiple organisms, their abiotic environment that acts as a single functional unit. Whereas a habitat is something that is best suited for a certain population to survive in. When we talk about population are a group of microbes. They are of the same species. Whereas when multiple species coexist, we call it as a community. So this concept is the basic of any ecological study. So be clear with this. And yeah, all of you have answered it right. This is a third question. Number of different kinds of organisms or types of species present in a particular area defines. That is not biodiversity. Here you have to look in to the term that I have stated different kinds of organisms or different types of species present in a particular area. Any other different answers? No, it does not define community. Community is something where different species do coexist or live together, but this does not define community. Ecosystem. Ecosystem together will also consider about the abiotic factors also, not only depending on the species of the organisms or the biotic factors in that sense. This is one of the parameters of the ecological studies. So the number of different kinds of organisms or types of species present in a particular area defines the richness of that area, like how rich the biodiversity is. We have seen about the operational taxonomic units, or the number of species that we can find out from a particular ecosystem or a point of sampling where we have considered. So in that, uh, the different kinds of species that we see, that defines the richness of that area under sampling, right? Biodiversity is in a very general context wherein you mean about different kinds of organisms, be it plants or animals or whatever, or microbes or whatever. But when you are going to consider either their species level, genus level, or other classificational level organization, then you call it richness. So, next question. Evenness is a measure of dash of the different species making up the richness of an area. You can try answering this question based on your understanding. Two people have answered. I'll wait for some more answers and then discuss. Any more answers? Okay, relative abundance is right. So evenness is a measure of relative abundance of the different species making up the richness of an area. It is not uniformity because like uh, when it is more uniform, there is no need of any ecological study, or it doesn't bring about to any research question uh, regarding to how the chemistry works, how the microbiology of the system works. Okay, it is all going to be well ordered, and there is not going to be much of questions to be studied in that kind of an environment. But we have to study about the relative abundance. Relative abundance is very important because how the presence or abundance of one group signifies certain characteristics of that particular ecosystem or the environment uh, in relation to other species that are present there. That is what we study in ecosystem, the interrelationship among the species, also their interaction with the other chemical physical factors that are present in the system. So we have to study how abundant one species is with respect to uh, the entirety of the population there. If there are 10 species making up that ecosystem, how the number or the chemistry of one is favored, and how that affects others. So this is the study of relative abundance. So you have to have this term in mind, that evenness always deals with the relative abundance of different species that makes up the richness of an area. So here we have a small problem. I'll give enough time for you to solve. I have given 7 OTUs, and this is the abundance of the OTU. I just want you to calculate the Simpson's index of diversity for the given problem statement. I hope all of you know how to calculate the Simpson's index of diversity. So here I have given you 7 operational taxonomic units and their abundance in any ecosystem that was taken for sampling. Okay, so with this data, I just want you to calculate the Simpson's index of diversity for this particular sampled area. I hope you people are trying. I'll give you some more time and see if any of you could give me the Simpsons index of diversity for this. Did any of you get the answer? Are you people working with the problem or are you stuck somewhere? Uh, how many of you have started? Okay, what will be the first thing after this that you would have found out? Any of you yeah, got started with the problem? Can I get some responses in the chat? Like, did any of you start with the problem? Yes, no. I see a thumbs up. I guess you have started working with the problem, right? Or what was the first thing that you found out after this abundance? Any of you could respond in the chat box. What will be the next thing that we have to find out here for Simpson's index of diversity? Okay, should I display the answer and explain? At least respond for this. If yes, give a yes. Should I display the answer directly and explain, or should I wait for you? Okay, one person wants the answer to be discussed. Okay. Okay, so this is the answer for that problem. So first of all, I have given what are the OTUs and what's the abundance. First step is you have to find the sum total. So which is 400 for this, and you have to find the PI value. Anyone can tell how you will find the PI value? This was explained in the lecture, no? Any of you can put in the chat box. Okay, I see no responses. That's fine. So PA value, how you have to find is you have to divide the abundance value by the total abundance. So 45 by 400, 50 by 400, 65 by 400, 55 by 400, 40 by 400, 50 by 400, and 95 by 400. That is the PA value. So you have to find the PA squared value, just square of this term, and find the summation of sigma pi square. So sigma pi square is the Simpson's index. Okay, the d value. This gives the probability that the two randomly selected individuals in the community belong to the same category. It's the Simpson's index. When you have to find the Simpson's index of diversity, you have to subtract Simpson's index from 1, which will give you this value. Okay, so this index of diversity is the probability that when I'm going to select some random individuals from a community, they belong to different categories. Okay, so I'll explain it one more time. I've given you the abundance of each of the OTUs. What you have to do is you have to first add up everything, and you have to find the PI value. To find the PI value, you just have to divide this individual abundance by the total abundance value, and you will get the PI value for each of the OTUs. And after that, you have to so your Simpson's index will be the summation of PI squared. So for that, you have to find the PI square and add up everything together. This will give the Simpson's index d value. Okay, this is the d value, this 0.156. When you are subtracting it from 1, you will get the Simpson's index of diversity. The difference between Simpson's index and the Simpson's index of diversity is that Simpson's index will give you the probability or that two randomly selected individuals in the community belong to the same category. Okay, as the name suggests, the index of diversity is going to give you the probability that they do not belong to the same category, but they belong to different categories. Is this clear? If you want me to repeat, just give it in the chat box so that I'll repeat it one more time. If it's clear, also just let let me know in the chat box. Okay. So here I have asked you to calculate the Simpson's index of diversity for the given sample given sample. Okay, so in my sample I have found out 7 OTUs, operational taxonomic units. Okay, and the abundance of each of the OTUs was also given. So in order to find the Simpson's index of diversity, so the formula for Simpson's index of diversity will be 1 minus capital D. Okay, so for that you will first need to find out what is D. D is equal to sigma pi squared. Okay, so in order to find this d, you need pi squared values. So for that, you will need the pi values. How to find the pi values is you just have to divide um, the abundance value of the first tax OTU by the total abundance. So 45 by 500 will give you 0.113. Similarly, 50 by 400, 65 by 400, you have to divide each of this abundance value with the total abundance, that is 400, and you will get the PI values here. Okay, so when you are going to square this PI value, you will get the PI squared, add up everything, that is sigma PI square, which will be your D value or the Simpson's index. So this 0.156 is the D value, which tells you the probability that two randomly selected individuals from this sampling that we have done belong to the same category. Okay, but this was not the question that was asked. I asked for Simpson's index of diversity. So for that, what you have to do is just subtract this 0.156 from 1. Uh, in probability you have studied, if the probability of success is P, probability of failure will be 1 minus P. The same way, I have asked for what will be the probability that the selected individuals do belong to different categories. So here you have got the probability that they belong to the same category. So when you're going to subtract it from 1, you're going to get the Simpson's index of diversity, which will tell you that when I'm going to pick up two individuals from my sample, what is the probability that they belong to different categories or different OTUs out of this 7. So that will be 0.844 for the given problem statement. Is that clear? Is this clear? Okay, I see someone raising your hand. If you have any doubt, you can post in the chat box or you can unmute and ask. Okay, I guess this is clear. I'll post the next question. I hope you are also clear with the definition of this index, Simpson's index and the index of diversity. They are just 1 minus Simpson's index will give you the index of diversity. Okay, have that in mind. And read the question carefully, whether they have asked for Simpson's index or Simpson's index of diversity. When asked in your MCQs kind of a question, such big problems may not be asked. It can just be like they will give some most of the values here, or they can at least give you the Simpsons index and ask what is the Simpsons index of diversity, or vice versa, being it multiple choice or multiple select question. You can get problems like that also. So I'll post the next question. That is also a problem. I'll give you enough time for you to solve this. This is the next index that was discussed, the Shannon-Wiener index. If you have any doubt, you can unmute and ask.
Um, Ma'am, I have a doubt from assignment 4. Can I ask it?
Okay, yes.
Ma'am, the question was that disinfection kills all the microbes. True or false? Ma'am, what will be the answer?
True.
Ma'am, disinfection kills all the microbes.
Yes.
Shouldn't it be sterilization?
Disinfection disinfection, see, you will be in case of water treatment, when you are asked to disinfect the water, you are not going to give any specific treatment that is going to just target your infective microorganism and kill it. Okay.
Sorry, ma'am, couldn't understand, ma'am.
uh, Sterilization, in the case of like media sterilization or something like that, technically use the term sterilization, where you also kill the fungal spores, everything together, right? Like autoclaving, etc. You're talking about that, right?
Yes, ma'am. Ma'am, disinfection can't kill all microbes, right? Spores are not killed by disinfection. Sterilization is used for that purpose, na?
uh, spo-
And
Spores are not microorganisms. They are specialized bodies that the microorganisms use to survive extreme conditions, like, or unfavorable conditions for that matter. But in the case of disinfection is what you do in the case of water and wastewater treatment, right?
mhm.
When you are
Ma-
going to talk about the scope of this particular course, you are not talking about killing microbes in general, for a particular purpose or something. It is just about going for a process that gives you a clean water. So
ma'am,
okay,
oh,
yes.
ma'am, we are microbiology students, so we will consider disinfection and sterilization as different things. So we thought it will be false, but that's why we are not able to understand why disinfection is considered like true. The statement is true.
Okay, okay, I get your doubt. See, mm, in terms of microbiology, disinfection will mean only killing of infectious agents, right?
Yes, ma'am. Only
uh,
vegetative cells, not
Only
the spores.
vegetative cells, not the spores. But in case of water and wastewater treatment, which comes under the scope of this course, disinfection, generally we use chlorination or ozone treatment or UV light for that matter, to kill the microorganisms that are present in the water.
Okay, ma'am. I got it now.
So that's a basic difference. In this context or the scope of this subject, disinfection will cover killing of all the
Uh,
microorganisms.
okay, ma'am.
Okay,
Thank you, ma'am.
you're welcome. I hope you people are solving. At least you can find the PI values. Abhishek Das, I think you are asking the bond between pentose sugar and the nitrogenous base, right? It is a glycosidic bond.
No, ma'am. The question was this only bond between pentose sugar and nucleoside. It was a question in our assignment.
Okay.
Ma'am, the bond is glycosidic bond, but we answered it as false because question तो थोड़ा अलग लग रहा था.
It is.
Ma'am,
It should have been a nitrogenous base, not a nucleoside per se. You can raise this question in the discussion forum so that the course TA will respond to you. But as far as the concept is concerned, the nitrogenous base and the sugar are linked by a glycosidic bond.
yes ma'am, that's why we were confused कि मैम ये आंसर क्या होगा कि न्यूक्लियोसाइड में ही ग्लाइकोसाइड होता है एंड ये पेंटो शुगर एंड ग्लाइकोसाइड पूछ लिया है.
Uh, might be a error with the question only. Okay. Oh, so your submission deadline is also over, and even I am not allowed to discuss your um, questions before you your deadline is over. So if you have any things issues with the questions like this, you can just post it in the discussion forum only so that the respective course TA will take up the questions. You can still raise this issue so that if the question is wrong, like I think they can give you marks for all of you. Okay, as far as I see, the question should be the bond between pentose sugar and a nitrogenous base. Because nucleoside in itself contains the base and the sugar. Sure, sure. You ask in a discussion forum. Not much. This is a 12-week course, so you have more of assignments. Your score will not be affected much. Anyone who has found out the Shannon-Wiener index for this problem? Anyone who knows how to proceed to find the Shannon-Wiener index? I think I have explained it for Simpson's index, right? It's going to be something similar but with different significance. So, anyone who has an idea how to find the Shannon-Wiener index? Okay, let me give me a thumbs up if I have if someone is trying this out and I should wait for them to respond. Okay, how many of you at least found till the PI value? Okay, after PI value, what any of you know what should be done next after finding the PI value? I think this was given in your course content this week. It's just a practice example problem for the concept that was discussed. Okay, so shall I display the answer for this question and discuss? Okay, I'm giving the answers now. So for the Shannon-Wiener index, the formula is sigma of pi ln of pi. Okay. So first of all, as told, your abundance is individual abundance value for each of the OTU is given. So you can find total was also given. If not, you can find the total, and then you have to find the pi value by dividing the abundance with the total abundance value. Sorry, dividing the abundance value with the total abundance value, you can find the individual PA value for each of the OTUs that is given, and you have to find the natural logarithm of uh, using this PI value for the individual OTUs, and then you have to multiply this PI and the ln PI value. So you get multiple values. You when you find the sum of all of it, you get the Shannon-Wiener index. Is this clear? I'll repeat it one more time. The OTU value and the abundance value was given. All you have to do is divide the individual abundance value by the total abundance value to get the PI value. So then you have to find the natural logarithm of the PI value and then multiply PI and ln PI value. When you are going to find the uh, when you're going to find the summation of pi ln pi and take the negative minus pi ln pi will give you the Shannon-Wiener index. I think last time I forgot to mention this. You have to find the negative of sigma pi ln pi. Is that clear? Is that clear? Okay. So as far as I have seen, I guess when there is some numerical problems, you people get lost. Try so from the next session in the next session also I'll be including some numerical problems either from that week's course content or the previous content. You just try and at least be in a state that you move towards the right direction. So in this, if you have at least found out this pi, ln pi and stuff, it's good. So finally you can just have remembrance of the formula and then find the index value. Okay, so be careful with the numericals. Try to observe how to approach for problems. So here is a theory question next for you to answer. I'll wait for a couple of minutes. Okay, so answer for this question as answered by Nikita correctly is A. Uh, so NAD plus and NADH redox pair has the lowest redox potential. If you remember the redox tower that we have been discussing for so many weeks now. So in that you can find uh, the oxygen water redox pair has the highest redox potential. So that is why oxygenic microorganisms are highly energy efficient compared to other sources. Okay, and the components of the electron transport chain are organized in terms of their redox potential. Yes, and then the redox potential of a system is usually compared with the potential of hydrogen electrode. So the whenever you tell about this redox potential, you come across a term called the E naught value or the E value. So this E naught value or E value is a reference value. So this there is nothing absolute about the potential. So the potential has to be described using some of the other scale. In general, the scale that is used for describing your E value or the E naught value is the hydrogen electrode. It is considered to have a zero potential, and based on that, negative potential or a positive potential is given to individual systems. Any doubts in this question? Also here you have to be careful regarding the pairing. When it if even if it is given when the pairs are changed also the answer will be wrong only. A redox pair has to be read in this particular notation only. NADH and NAD plus redox pair, uh, you cannot read it in that way. You have to read it as NAD plus NADH redox pair has the lowest redox potential. So that's the answer for this question. Next question, match the following with respect to the terrestrial environment. You can give the answers as A1, B2, something like that in the chat box. Any answers? Even if not all four right, whatever you know matches here. Can I get some responses? more responses. There is one response. It's A4, C2, B1, and D3. I think you are right. So yes, here is the answer. A horizon refers to organic matter accumulation. Okay, and the B horizon is where the organic matter depletion takes place. O horizon is generally the topmost where you have this organic debris or the for example leaves, litters, etc. So that's the topmost layer, and the R horizon is the bottommost. Okay, this is the hard bedrock. So generally in terrestrial environment, you also have the C horizon, but as far as this question is concerned, it's O, A, B, and R in the order, and A constitutes organic matter accumulation, B constitutes organic matter depletion, O is for organic debris, and R is for hard bed rock. I think Shavufta got all of it right. Good. So this is the next question. Which of the following is non-photosynthetic? I guess this question is not that tough. Okay, why? I don't know why are you confused more. Which of the following do you think are completely photosynthetic? Or which option is confusing here? Any of you, can you respond? Like, what is the confusion here? You can also type two responses between which you are confused. Any of you would like to take a call? Okay. So I'll display the answer for this. The answer for this is methanosarsina. See, okay, I'm confused between A and D. Uh, I think almost all of you are pretty sure that cyanobacteria is photosynthetic. Okay, diatoms are also algae. So basically, they undergo photosynthesis. When it is confusing between A and D, see green sulfur bacteria. Uh, Usually, when there is some pigment or color associated with the bacterium, it is not simply some decorative element or something. It will have its own metabolic significance, right? So when you find some color attached to it, you can just when you're out of idea, you can just guess that the color associated will be due to some pigment, and the presence of that pigment will obviously have some connection with light. So it is going to have a pigment in order to sequester some kind of or harvest some kind of energy from the surroundings, basically light. So in this case, methanosarsina, if you are aware about methanosarsina, it is one of the methanogen. So it generally generates methane. So when you are going to generate methane, it has to feed on some carbon source. It is not going to take up carbon dioxide and build upon it and then generate methane. Okay, so that is one way of answering is, if you know about methanosarsina, you will directly tell that it's a methanogen, so it's going to be uh, non-photosynthetic. When you have such options and you are asked to go for an select one out of it, you can just think in this way also, like when I am specifying some color for this bacteria, it is obviously going to use that color for harvesting light for some or the other purpose. So we know that in our case, it will generally be harvesting light for synthesis of some compounds only. So that way also you can answer this question. And the answer is methanosarsina is non-photosynthetic here. Is that fine? Okay. Okay. We'll solve a few more easy questions. I think most of the questions from this onwards will be true or false. This is a true or false question. You can just type in whether if you think that statement is true or false. Okay, any of you tell me what is ORP? Right, oxidation reduction potential. Yeah. So now tell me whether the oxidation reduction potential determines functional class of microorganisms in an environment. This particular concept has been discussed for so long now in the lecture videos also. Any guesses? Any guesses would do. Okay, we have studied about classification, different classifications of microorganisms under based on like different concepts, right? So when we studied about this microbial nutrition, what were the parameters that we considered? That is one way. Uh, Regarding that sources of nutrition for this microorganism, what and all did we consider? We discussed one flow chart. Okay. See, uh, did we discuss about carbon source, energy source, electron donor, electron acceptor, and stuff? I guess you remember now. Ah, right. So we have discussed about electron donors and electron acceptors, right? So what are these electron donors and acceptors? So based on the potential that is present there only, we can classify which can act as a donor, which can act as an acceptor. Uh, why? What is the significance of this electron transport or the electron transport chain or the electron transport system? All of this we have discussed. So the oxidation reduction potential determines the functional class of microbes in an environment. Obviously, you want to give a very simple example. uh, I think in many of the examples it was quoted that oxygen will be present in the topmost layer, and then you will have sulfate reducing bacteria, sulfur reducing bacteria, etc. Right? All these are determined by the oxidation reduction potentials, like whatever nutrient that can be utilized by the microorganism for um, electron donation or electron accepting. Both this electron donation as well as accepting happens, only then this redox reactions happen. And as said, all or almost all of the biochemical reactions will involve these redox reactions only. So oxidation reduction potential is an important factor that determines the functional class of microorganism in an environment. When you have an oxygen rich environment, you will have the aerobic organisms, and then you will have nitrate reducers, sulfur reducers, uh, all organisms like that. And finally you will move to anaerobic environment where there is no oxygen present. Okay, so this oxidation reduction potential is a very important factor that determines the functional class of microorganisms. So the statement is true. So this is the next question. Very easy question. Then what is the term Shabutakan? Okay, this statement is true. Like converting organic sulfur present in your organic matter to hydrogen sulfide is actually desulfurization. Next question. This is also a true or false question. Nikita, do you have any explanation of why you think it is true? You can even unmute your mic and tell if you feel it's very long to type. Okay. So, what is succession and what is evolution? Changing condition. Yes, the change in the microorganisms in any particular environment with a changing condition is called succession. Okay. Um, Both succession and evolution happen with respect to time only. In succession, what happens is like one microbe succeeds the other microbes because of some advantages that it has in order to thrive in that particular environment. Whereas in evolution, there is a modification or a change in the organism itself, maybe a functional modification or a structural modification, something like that happens in evolution. So in this statement it is false because microbial communities undergo succession. That is a different uh, perspective. uh, it it has nothing to do with evolution. um, for example, when you consider microbes only, maybe I have anaerobic species living in my environment. uh, Maybe after some time, because of some biochemical reactions, as the earth formed also, there was presence of oxygen, and then all these aerobic organisms or oxygenic organisms were able to survive. This These microorganisms are said to be succeeding one another, and the a microorganism that was initially present will be called as the predecessor. The one that follows will be called as a successor, right? This is what the concept of succession is about. In case of evolution, in order to adapt to a particular changing environment or something, there is some modification that is happening in the organism. If there is a modification happening in the organism, that phenomenon will be termed as evolution, and evolution has nothing to do with succession in all the cases. They are interrelated, but it is not always necessary that in order to undergo succession you will be needing evolution. Evolution may take even more longer than this succession phenomenon. So this is a common question. Just to remind you of that term, all ferric reducing bacteria can form a or all sulfate reducing bacteria can form a, whatever the question is, the answer is going to be the same. We have discussed about population, we have discussed about community in similar context. No, Shagufta Khan, it will not be a population. In population, you will have only a single species, right? Whereas there can be multiple species of ferric reducing bacteria. What will you call them in that case? Not a community. Community can include different bacteria like ferric reducing, sulfur reducing. They need not have the same function. They need not belong to the same species also. They do not have same similar function also. They are based on the function. What can we call? This is a very simple question if you have gone through the lecture videos, lectures basically. But the term is very important. Any of you have a guess? It is not population, it is not community. It can be either all ferric reducing bacteria or all sulfate reducing bacteria or nitrifying bacteria. Okay, so I would recommend you all to go back to the lectures once again. When we are going to classify the microorganisms based on their function, all the similar functionality function performing bacteria or microbes together will be called as a guild. They necessarily need not belong to the same species, but they perform the same function. So that is why they form something called a guild. Okay, right. Generally, what are biofilms made of? What do we call the layer? Right, I have got two responses. Okay, as you have told, it's true. Biofilms can give protection to the microbes in harsh environment. They are help all for the adherence of the microbe. Basically, they form films. They adhere to it so that to give them advantage in terms of acquiring nutrition. And as rightly told by Shagufta Khan, it's EPS. EPS stands for extracellular polymeric substances. So they are basically polymers, carbohydrates, proteins, etc. To which this microbe secretes themselves, and then they get attached to them uh, to get some nutritional advantage. Something these biofilms can also give protection to the microorganisms in terms of harsh environment. I think this is the last question for today. With this question, we'll wind up. Right. So higher the BOD, higher the chances of water to be contaminated with contaminants such as sewage. Yes, it's true. BOD is basically the biochemical oxygen demand, which is the oxygen required that will be required for depletion of the organic matters biochemically. So it is a indication of pollution. When the BOD is higher, the chances of contamination or pollution or the utilizable organic matter content is more. This has to be reduced by using treatment techniques, and then finally it will be fit for different purposes, right? So thank you all for joining in this session. Um, though this was a postponed session, I highly appreciate your time and your presence and participation. Thank you for joining. If you have any doubts, you can ask. Otherwise, you can leave the session and we'll wind up. And as told, this session was postponed because of some health issues. I was not able to conduct the session on Tuesday. Next session will happen on Tuesday only. So all the next sessions, all the other further 8 week sessions will happen on Tuesdays only. So if you have any doubt, you can ask. Otherwise, you can leave the session. Thank you for your time and your active participation. Thank you.